Hi everyone, this is Nehad from Bio with Nehad channel. In this video, we're going to explain the nucleic acid. We started the biochemistry before. We have been talking about carbohydrates and lipids in the previous two videos. But this video, we're going to explain the two types of the nucleic acid, the structure and the function of each one of them, and we're going to compare between them. Let's get started. We have been talking about the biological macromolecules. We said it's biological because it's in the human body, and macromolecules because it's actually a big molecule, each of them. And the point is they are called polymers as well. What the meaning of polymers? That it's made of small monomers, small units. They are connected together by a bond. So our video today, we said it's about nucleic acid, and the monomer is the nucleotides. So the nucleic acid as a polymer, it's made of small polymers, monomers, and the monomer today it is called the nucleotide. The nucleotides are connected by those bonds to give the long strand of the DNA or the long strand of the RNA. Here we have the structure of the nucleotide. It is composed of three units. Number one is the sugar. It's a pentose sugar. Pentose, you see here a pentagon. So like related to the pentose. So 1, 2, 3, 4, 5. But actually it is called the pentose because it is composed of 5 carbons. Okay, so a 5 carbon sugar, a pentose. The second unit is the phosphate group connected to the sugar. It's the phosphate group. And the third one here connected to the sugar as well, the sugar molecule, it is the nitrogenous base. Nitrogenous base. So number one, sugar. Number two, phosphate. Number three, nitrogenous base. And this is the real structure of the nucleotide, the phosphate group, the pentose sugar, and the nitrogenous base. We're going to explain now the structure of the DNA molecule. I have two strands in the DNA molecule. I'll highlight, this is the first strand with the green highlighter. Okay, so this is one strand, and this is the second strand. The first strand, or both strands, they are made of nucleotides, as we said, and they are connected together. So this is the nucleotide, one of the nucleotides. Phosphate group is here, the 5 carbon sugar is here, the ribose sugar or the deoxyribose in the DNA, and this one is the nitrogenous base. They are connected together by this covalent bond. So a covalent bond, another covalent bond, another covalent bond, to just connect all of them together to give that strand. Then to connect the two strands together, we have here the hydrogen bonds to connect the nitrogen bases of the two strands to give a molecule made of two strands. So those are the two strands. Those are the two strands, and they are connected with the hydrogen bonds. If you notice that this one, the direction of this one from 3 to 5, and this one from 5 to 3. So one of them is like upside down or like inverted. Okay, they don't have the same direction, they have opposite directions. I'll just clear what I did so I can give you more information. So for those, they said that the structure of the DNA, it's more like a ladder. So the ladder is so I'll just show you the phosphate here, the phosphate sugar backbone, they are calling it phosphate sugar backbone on the two sides, and here in the middle we have the rungs of the ladder. Okay, so it's like so much like a ladder. The point is, when I heat up the DNA molecule, not all of these bonds will be broken down, only those in the middle, that they are connecting the nitrogenous bases together. Why? Because those are hydrogen bonds. The others are covalent. So the hydrogen bonds, they are weaker. So they are easier to be broken. So when you heat up the DNA, the first weak bond, this is the hydrogen bond, this one will be broken. In the nucleotides, we have the same structure. So the phosphate group is the same, the sugar is the same, the pentose sugar is the same, 5-carbon sugar is the same, but the nitrogenous bases, they are different. We have 4 types: adenine, thymine, cytosine, and guanine. So in the DNA, we have those 4 different nitrogenous bases, but actually the only difference in the nitrogenous bases that the RNA has, adenine again, guanine again, and cytosine. But instead of T, thymine, we have the uracil. Uracil, uracil. It's only in the RNA. So when you see a strand, one strand has uracil, has adenine, guanine, cytosine, but uracil instead of thymine, so this one is an RNA. If I have adenine, cytosine, thymine, and guanine, it is a DNA. And actually, the DNA, it is made of 2 strands, not 1 strand. But the adenine and guanine is one group. It's purines. And the pyrimidines, it's the cytosine, thymine, and the uracil. So basically, the adenine has to connect the adenine or the guanine, which is purines, and they have 2 fused rings. They have to be connected with 1 ring. So each 2 rings, they have to be connected with 1 ring. Do you know why? Actually, this is the question time. The answer is that for this distance between the 2 strands, it has to be constant. Okay, it has to be precisely the same. Why? To be in that shape. Okay, in this neat shape. So the rungs here, it has to be the same. That's why a purine is always has to be bonded with a pyrimidine. So 2 rings with 1 ring. So collectively they have to be 3 rings in each rung. Okay, and to be more specific, we have something called called the base pairing. The base pairing, that the basis has a specific pair. So the C has to pair always with G, and of course G with C. Between them we have 3 hydrogen bonds, okay? And the A has to pair with T always, and between them we have 2 hydrogen bonds. So purine with pyrimidine, and more specific, C with G, A with T. And if it's an RNA molecule, you have to change the T with U. So when you see the base pair A with U instead of C, instead of T, this is the RNA. So here we have T is paired with 2 hydrogen bonds with A. G with C, 3 hydrogen bonds. A, what do you think? Quickly. Yeah, so T with A, 2 hydrogen bonds. Then C with G, 3 hydrogen bonds. And as you see, all the rest, they are connected, the sugar and phosphate. 2 phosphate, they are gone, and 1 phosphate is connected to the previous nucleotide. Then C with G with C, 3 hydrogen bonds, T with A, and C with G again, 3 hydrogen bonds here. Okay, and this one 2 hydrogen bonds. And see here, just focus on what happens. 2 phosphate and 2 phosphate is gone, and this one is connected to the previous nucleotide, covalent bond. Again, okay, so did you see how it works? To sum it up, we have 3 differences between the DNA and the RNA. You can stop the video and just think about it, about the 3 differences. I just said them in the video before. So the first one is, this one is made, the DNA is made of 2 strands, and this one, the RNA, is made of 1 strand. The second one, the basis, it's adenine, cytosine, guanine, and thymine. For the RNA, we have again guanine, cytosine, adenine, but the difference is uracil. It's instead of the thymine. Okay, so instead of T, we have U, and it is connected normally with the A. The same case. The last difference, it's the sugar. So the sugar in the deoxyribonucleic acid is deoxyribose sugar. For the ribonucleic acid, it's the ribose sugar. That's why the DNA, it's deoxyribonucleic acid, and the RNA is ribonucleic acid. Question time again. Here we have 1 strand of the DNA. Can you draw the complementary strand of this strand? You can correct your answer now. So if you have here G, you have to have C, base pairing, okay? A with T, G again with C. Between G and C, C and G, we have 3 hydrogen bonds. Between A and T, or T and A, we have 2 hydrogen bonds. Make sure that you drew the 2 strands in opposite direction. This one from 3 to 5, this one from 5 to 3. And you connected the phosphate with the sugar with covalent bond. So just make sure that everything is right. Thank you for watching. Just subscribe, follow and like all the videos, and see you in the next video.